by the investigation of the entire complement of a specific type of biomolecule or the totality of a molecular process within an organism. This is called omics. In biology, the word omics refers to the sum of constituents within a cell. The omics sciences and technologies share the overarching aim of identifying, describing, and quantifying the biomolecules and molecular processes that contribute to the form and function of cells and tissues. Branches. There are many branches of omics sciences. Example, well-established fields include genomics, proteomics, metabolomics. Genomics considers the totality of the structure and function of an organism's genome, the entire set of genes, while proteomics focuses on the proteome, that is the entire collection of proteins in an organism's cells. Metabolomics is concerned with the metabolism and more specifically with the function and interactions of metabolic breakdown products or metabolites. Other branches of omics include transcriptomics, the study of the full complement of RNA in an organism's cells, and lipidomics, which focuses on lipids and pathways involved in lipid signaling. The interdisciplinary nature of the omics sciences is reflected particularly in the field of interactomics, which bring together biology and bioinformatics to investigate the relationship and interplay between proteins and other molecules and the significance of these interactions. Technologies and data management. Technologies that are paramount to omics sciences include various high-throughput automated biochemical assays and software and databases that allow for complex integrated analysis. In genomics, for example, next-generation sequencing and assays designed to detect genetic variations play an essential role in identifying and characterizing genes. Microarray analysis and RNA sequencing likewise serve a critical role in transcriptomics, while microassay, liquid chromatography, mass spectrometry are examples of technologies used for identification and characterization of proteins. Such high-throughput assays generate large amount of data, which can be used for modeling and other forms of analysis, but which also present challenges for data storage. Multiple databases have been constructed to house data from omics studies and to render the data publicly available. Examples include Cancer Genome Atlas, the International Cancer Genome Consortium, Proteome Exchange Consortium, Proteomics Identifications Database, and Human Metabolome Database. Impact and challenges. The impact of omics is most apparent in medicine. Sequencing of human genome, for example, has fueled advances in personalized medicine, in which decisions about disease prevention, diagnosis, and treatment are tailored to patients based on the information derived from genetic and genomic research. In particular, genomic data have played key roles in the development of predictive models of disease and in informing therapeutic decisions, such as in the treatment of cancer. Similar links between omics and personalized medicine have emerged from metabolomics with the discovery of new biomarkers of disease. An example is the investigation of disturbances in metabolic pathways that affect levels of substance such as fatty acids and bile acids. This work has led to identification of biomarkers with the potential to improve early diagnosis of hepatocellular carcinoma. Nonetheless, significant challenges remain in the omics sciences, especially concerning the data complexity, data management, and the integration of data from omics studies with data from other sources, like clinical data gathered during routine physician visits. Other challenges are more fundamental, such as in assay development and refinement. In large-scale proteomic analysis, for instance, agents designed to bind to specific proteins often are lacking in sensitivity and specificity, decreasing their affinity for the proteins of interest and resulting in suboptimal protein capture. That is all about omics. Abstract. Cutting-edge omics strategies are bringing new opportunities to many fields of investigation, from biomedical to biomedico-legal. Comprehension of molecular mechanisms associated with pathologies such as acute or chronic toxicity or sudden cardiac death and discovery of associated biomarkers represent key elements in the development of novel routes for a better understanding of complex phenomena. Omics and mining of complex generated data have reached a state of maturity. Recent innovations, notably for mass spectrometry, with a remarkable gain in sensitivity and selectivity, and the development of next generation sequencing technologies have considerably increased the power of these approaches. In spite of ongoing progress in the omic methodologies, their application can already provide informative results leading to more accurate interpretation and evidences. Beyond the remarkable potential in forensic toxicology, such as research of new biomarkers, we predict that these technologies will reinforce the translational research between forensic and clinical disciplines. The development of omic sciences and their recent integration are bringing new insights to many areas of research. Multi-omic investigations aim at studying the overall changes and associated mechanisms of complex traits occurring in response to system-level perturbations. With the development of cutting-edge technologies and computational tools, a state of maturity has been reached, offering the possibility of overcoming the complexity of life sciences. Beyond the clinical application, such tremendous opportunities are expected to have considerable impact in the biomedico-legal field. Interestingly, forensic sciences are built on the subtle association of researchers with multiple skills and knowledge, integrating heterogeneous and synergic backgrounds. Many forensic institutes already integrate physicians, geneticists, toxicologists, chemists, pharmacists, biologists, and biostatisticians. That is, combining of all the competencies required in a system-level investigation. అది ఓమిక్ అంటే ఇన్ని కలిపి అనమాట, అన్ని కలిపితే వాడదు అది ఓమిక్ టెక్నాలజీస్. In a modern laboratory of forensic toxicology, mass spectrometry plays a central role for detection, identification, and quantification of pharmacologically active and or toxic substances in various complex biometric matrices, which may be involved in death cases, that is direct or indirect, and may affect the behavior or deteriorate the physical and mental capacities. Among these procedures, toxicological screening is usually one of the major steps throughout the systematic toxicological analysis. By definition, screening procedures should ideally enable the detection and identification of thousands of molecules and the related metabolites potentially present in complex biological samples. The use of peak detection algorithms, MS fragmentation, and library searching tools are different steps of the process allowing the detection and identification of molecules of interest. Although the purpose is totally different, the concept behind toxicological screening overlaps in certain points, the definition and methodology of metabolomics in certain points. At least from a technological point of view, metabolomics is the science aimed at characterizing and monitoring the broadest range of metabolites in biological matrices. The recent advancements and innovations in mass spectrometry in terms of sensitivity and resolution have driven the development of metabolomics by overcoming technical limitations associated with the complexity of this field. The integration of metabolomic strategies in the bioanalytical research pipeline has created new opportunities to better understand the molecular mechanisms involved in physiological and pathophysiological states. The methodology has been successfully applied in numerous areas, non-exhaustively, including cancer, cardiovascular diseases, obesity, diabetes, neurology, and plant biology. In the population-based cooperative health research of Augsburg, 140 metabolites have been quantified for 4,297 participants, and the level of several metabolites were meaningfully altered in pre-diabetic individuals. Using metabolite protein network and targeted approaches on serum samples, 7 T2DM related genes associated with these metabolites were identified by multiple interactions with 4 enzymes, among them glycine and lysophosphatidylcholine. Metabolites have shown to be strong predictors of glucose tolerance, even 7 years before disease onset. Other serum metabolites have been identified as predictors of diabetes in European prospective investigation into cancer and nutrition. More recently, Wang, using longitudinal data and 201 type 2 diabetes mellitus converters, identified a signature of 5 metabolites for which individuals in the top quartile exhibited a 5-fold higher risk of becoming diabetic converters. Because of its capability to detect slight changes in large data sets, metabolomics is also impacting the field of toxicology. In the last couple of years, clinical and forensic toxicologists have indeed shown increased interest in applying this approach to determine signature of exposition, susceptibility, and toxicity, for instance, early signature of hepatotoxicity, and to investigate, in relation to the observed molecular patterns, the cellular mechanisms leading to adverse effects of toxic agents and to the development of associated diseases. Interestingly, the comprehensive nature of metabolomics can also be used to simultaneously identify products of drug metabolism, such as reactive metabolites involved in toxicity mechanisms. In a pioneer study, Nielsen performed untargeted metabolomics of retrospective forensic cases of humans exposed to 3,4-MDMA to determine associated metabolites as well as endogenous changes related to drug response and toxicology. From whole blood samples collected over a period of 2 years, their retrospective study revealed significant relative changes of several metabolites, including known metabolites of MDMA and different endogenous metabolites such as acylcarnitines, amino acids, and tryptophan. In addition to the possibility of identifying new direct or indirect biomarkers of drug abuse from human data usually not available, this work confirms the interest of the approach for providing new insights into drug effect and toxicology. The changes in metabolite levels that can be measured by metabolomics will reflect the end response of biological systems to genetic or environmental modification. The ultimate goal is therefore to link these changes within biochemical pathways to the enzymes involved and then to the underlying genetic alterations. Development of multi-omic approaches is the next step to better characterize and understand a complex pathophysiological state such as a system organization. For instance, integration of genome-wide association study (GWAS) and the metabolomic profiling revealed how genetic polymorphism influenced the human metabolome, providing strong association between the metabolic traits and loci for biomedical and pharmaceutical interest. The potential omic integration is still untapped and will expand in the near future to cover a deeper picture of the dynamics of molecular systems. Many areas will be positively impacted by the promise of the post-genomic area. Not only the identification of more accurate interpretation, we can expect that these tools will enhance collaboration by potentially joining efforts between different experts of Forensic Institute. In this chapter, we will discuss the promising impact that omics could have on forensic sciences, notably by using metabolomics. We also aim to introduce imaging mass spectrometry (IMS), an emergent approach on MS, for the molecular, that is protein, metabolite and drug imaging of tissue sections, providing new opportunities in forensic toxicology and pathology. Heading: Omic study of alcohol consumption. Alcohol is a social drug whose harmful use is estimated to be 3.3 million deaths each year. Chronic alcohol abuse is a major cause of liver-induced toxicity and leads to the development of many other diseases, including cardiovascular diseases, cancers, and neuropsychiatric disorders. Direct and indirect biological markers are used to determine alcohol consumption. Indirect markers, including gamma-glutamyl transpeptidase (GGT) and carbohydrate deficient transferring (CDT), are biological parameters for which a modification in the concentration may be caused by high and regular alcohol consumption. Direct markers, including ethanol (ETOH), ethyl glucuronide (ETG), phosphatidylethanol (PETH), ethyl sulfate (ETS), and fatty acid ethyl ester (FAES), demonstrates a great sensitivity and specificity with respect to a recent consumption and present the advantage of being detectable in the organism only after alcohol consumption. Some of them, like ETG, are easily incorporated into the hair, allowing the extension of windows of detection in order to assess chronic alcohol consumption. With respect to the burden of alcohol abuse and the number of related diseases, identification of novel biomarkers is of major importance to develop novel routes for better assessment of alcohol intake, prevention and therapeutic strategies, but also to underlie the mechanism associated with its toxic effects on associated disease onset and development. Recently, GWAS has identified common single nucleotide polymorphism (SNP) associated with alcohol consumption, increasing the catalogue of susceptibility loci and the understanding of genetic etiology of alcohol use disorders. Kutalik performed a GWAS study on 3 population-based cohorts, each based on more than 5,000 volunteers, to assess whether genetic factors affect CDT concentration in serum. Their study revealed 3 single nucleotide polymorphism (SNP) significantly explaining some of the variation in CTD measured in parallel within the different cohorts. In addition to genomic studies, transcriptomics thought of what appears to happen and refers to the identification and measurement of all transcripts of mRNA, non-coding RNA and small RNA expressed in a specific cell, tissue or fluid to capture its specific and complete gene expression. Transcriptome profiling has generally been actively by hybridization based DNA microarrays, but is being progressively replaced by the recently developed next generation sequencing NGS technology. NGS extends the conventional process of capillary electrophoresis based sequencing across millions of fragments in a massively parallel fashion by means of a generation cluster from a library prepared by random fragmentation of cDNA sample. NGS is a powerful and cost-effective tool for ultra-high throughput transcription analysis, thus providing a discovery tool untapped in forensic sciences for deciphering the regulation and networks of gene expression. An example, Faris performed a RNA sequencing study on post-mortem human prefrontal cortex to investigate the perturbations in gene expression profiles and their network organization in alcohol-dependent individuals. Their work revealed association of multiple ion channels, including human-specific isoform of the voltage-gated sodium channel subunit SCN4B, underlying lifetime alcohol dependence. Other omics strategies have been applied to assess biological processes in development of drug abuse and the research of potential biomarkers. Among these approaches, proteomics has been widely applied, and Wang listed 237 proteins meaningfully associated with alcohol exposure from 17 proteomics studied. In a population-based cohort study, targeted metabolomics was also performed to investigate potential correlation between alcohol intake and changes in serum metabolite levels and to identify potential biomarkers that could predict high level of alcohol consumption. This study allows the identification of metabolic signature including lysophosphatidylcholine, either ether lipid and sphingolipid species that differentiate between moderate to heavy and light drinkers, and which was further replicated in other cohorts. Although the study was based on a metabolomic strategy targeting 131 endogenous metabolites, the data suggested new insights into the impact of alcohol consumption on human metabolism and demonstrated the possibility of revealing new biomarkers. Analysis of gene polymorphism, gene expression, protein metabolites and their potential combination is necessary to find key players with the largest detectable effect on a pathophysiological state such as alcohol abuse. This argument underlies the importance of integrating network information from multi-omic approaches. Taken together, these examples should encourage new study in different areas of forensic sciences to substantially improve our capacity of investigation at an unprecedented scale. Next heading, proteomics and metabolomics imaging by mass spectrometry, a link between forensic toxicology, pathology and radiology. In the last year, radiological imaging techniques have been remarkably developed and implemented in forensic institutes, especially in Switzerland, including multi-detector computed tomography (MDCT) and magnetic resonance imaging (MRI). The advantage of these approaches are undeniable by providing digitalized data to the forensic scientist that can be used in court or post-processed over time according to expert requirements. The development of imaging procedures will continue to grow in forensic sciences by taking on increasing importance in case studies. This phenomenon is due to the basic principle that complex set of data can be more easily understood from a single image, worded by adage, a picture is worth 1,000 words. In the last decade, the innovative MS-based approach has emerged for direct molecular imaging of tissue sections. In addition to MS efficiency for monitoring and identifying molecules, imaging mass spectrometry (IMS) leads to information on the spatial location of analytes in thin tissue sections. For the first time, IMS enables the simultaneous mapping of untargeted profiles of hundreds of molecules, including proteins, peptides, lipids, drugs, metabolites, while maintaining a high correlation between the resulting molecular images and the histology of the sections. Although the principle has been comprehensively explained elsewhere, the concept lies on the use of ion or laser beam to raster a thin tissue section mounted on a microscopic plate according to a grid array of fixed resolution defined by the user. At each coordinate, usually between 10 and 200 micro μm, a mass spectrum will be acquired, allowing the generation of ion images of tissue sections for each detected peak. The possibility of carrying out proteomic and metabolomic imaging from thin tissue sections is bringing new insight into the understanding and diagnosis of diseases. During the last decade, IMS has been applied in various fields like cancer research, cardiovascular disease, neurosciences, drug metabolism and distribution, plant sciences. In addition to the technology is also gaining in popularity in the field of forensic science. As an example, chemical imaging of latent fingerprinting were recently demonstrated, providing an associated chemical information for the physical identification of individuals. Although IMS has been extensively applied to the analysis of endogenous molecules, IMS of drugs and metabolites is being more deeply investigated in recent years. As for instance, in preclinical studies for monitoring the distribution of drugs and metabolites in organs. As a remarkable example, IMS has been applied for mapping drug incorporation from a single hair analysis. In a recent study, the use of IMS provided crucial information by visually demonstrating that methoxyphenamine is incorporated at two different sites, that is the region of hair bulb and sebum gland. The implementation of IMS will also bring research opportunities in forensic pathology. A recent publication has demonstrated the interest of IMS for the profiling of protein in postmortem human spinal cord samples obtained from amyotrophic lateral sclerosis. In our institute, we are applying proteomics and metabolomics IMS in order to determine molecular changes occurring in the context of early myocardial ischemia and infarction. EMI ischemia during the initial 4 hours, which includes lack of specific symptoms, markers and tissue morphology as well as timing of markers appearance. Another emerging area is the field of development of 3D IMS to provide spatial molecular information of tissues or organisms. Indeed, 3D reconstruction of the multiple 2D IMS datasets presents an opportunity for a deeper study of many biological phenomena such as cardiovascular disease and neurological disorder. Despite numerous technical challenges still to be overcome, the method is highly promising and has already been successfully applied. In a recent study, we demonstrated the potential of the approach in both mouse model and human carotid atherosclerosis tissues to describe 3D biological system. Multiple concerns about the representativeness of the sample. Interestingly, 3D IMS has also been integrated to MRI, enabling the correlate postmortem proteomic imaging with corresponding in vivo anatomical data provided by radiological imaging. Looking at the growing importance of postmortem imaging, notably for the investigation of cardiovascular pathologies, integration of IMS to radiological methods into multimodal strategies will extend the potential of investigation by allowing the molecular information to be associated with anatomical features. From a general point of view, IMS is offering exciting new possibilities in the forensic toxicological field to obtain completely new information. Conclusion. Implementation of the omics strategies in forensic sciences is opening exciting novel routes to investigation. The power of omic methods and the possibility of integrating multi-omic approaches will bring new opportunities in the future to better understand complex phenomena and extend our capacity of investigation. The plethora of technologies that have reached a mature state of development now allowing for the in-depth analysis of genome, epigenome, transcriptome, proteome and metabolome. Although the future is promising, efforts are still required to push other technologies forward, especially from the bioinformatic point of view, where the computational resources still have to be improved so as to fully take advantage of tremendous quantity of the data generated for analysis. This is among the most important aspects for which the community has to unite in their efforts. This is J.R. Hunter. Although will never replace conventional forensic investigation, omic research has to be promoted to bring complementary tools. A way to move in this direction is also to accept that such an implementation represents an investment and may need time to deliver concrete application. Recent success stories concerning omic applications in biological clinical studies should encourage the user of these strategies in legal medicine. Elucidation of forensic cases is often a complex task overcome by the convergence of multidisciplinary experts working closely together, including physician, a geneticist, a toxicologist, a chemist, a pharmacist, a biologist, and biostatistician. All the elements are combined to foster the development of omic approaches in the field. These approaches have been already successfully applied in forensic toxicology with, for instance, the use of metabolomics to determine the signature of drug exposition and toxicity, bringing new opportunities to determine novel biomarkers of drug exposition, susceptibility, and toxicity. Enriched by a decade of remarkable developments, IMS has also witnessed a phenomenal expansion, bringing remarkable opportunities to develop new fields of investigation at the frontier between forensic toxicology, pathology, radiology investigations. The continuous innovation of omic strategies has considerably increased the power of these approaches. This gives the possibility of tackling very different issues in the new era of forensic biology. Among the potential future applications, we can note the understanding of various molecular events involved in pathology, such as acute or chronic toxicity or cardiovascular disease, and the research of associated biomarker. Recent literature indicates that the application of omic approaches in forensic research are only in the early stages despite remarkable potential. It seems obvious that their growing interest in legal medicine community will provide meaningful output, enhancing our knowledge and our ability to provide novel evidence in forensic sciences. Forensic sciences. Chapter is divided into 3 sections. Outlines the emerging role of forensic sciences and legal medicine in humanitarian action, which includes all interventions aimed at furnishing aid and assistance designed to protect lives and health, alleviate suffering, protect human dignity during and in the aftermath of man-made crises and natural disasters, as well as prevent and strengthen preparedness for the occurrence of such situations. All of the areas and sub-disciplines of forensic science and medicine can profitably be applied in the humanitarian forensic action. In particular, forensic anthropology, odontology, genetics, pathology, and clinical, legal action and forensic medicine. Forensic expertise in human rights protection serves 4 main purposes: to help families uncover the fate of their loved ones, to collect and document evidence of inhumane crimes, to use that evidence for convicting the offenders, and ultimately to set up new and more efficient prevention strategies. Introduction. Currently, a widely shared and or universal definition of humanitarian action is lacking due to the fact that the field of analysis, that is law, ethics or politics, influences the definition and principles behind this definition. Putting together the most important international bodies and associations such as UN, WHO, one could argue that a humanitarian action provides life-saving services and facilitates the return to normalcy for people in communities affected by natural and man-made disasters. It also seeks to lessen the destructive impact of disasters and complex emergencies. Humanitarian response is guided by the principles of humanity, impartiality, neutrality, and independence, which provide a common framework for organizations involved in humanitarian action. In this chapter, the role of forensic science and medicine in the humanitarian action is presented and discussed. It could seem a new field of application of forensic sciences, although, to say the truth, it is not really that new, as forensic pathologists and anthropologists have worked in this field since at least 50 years. The first systematic medical investigation of human rights abuses was used as evidence in an internationally recognized trial in Nuremberg for war crimes involving medical experimentation by Nazi doctors. After this contribution, there was a long pause in the involvement of medical science in documenting human rights abuses until the mid-1980s, when Dr. Clyde Snow intervened in Argentina with a team of forensic practitioners investigating the cause of death and injuries sustained by victims of the reigning military junta. Since this first intervention in Argentina, the forensic community has become more conscious and more knowledgeable about its role in recent times, having given it the designation of Humanitarian Forensic Sciences. Currently, there is no standard or legal definition of humanitarian forensic action, although it could be synthesized as the range of forensic actions furnishing aid and assistance designed to protect lives and health, alleviate suffering, protect human dignity during and in the aftermath of man-made crises and natural disasters, as well as prevent and strengthen preparedness for the occurrence of such situations or recurrence of such situations. The above definition includes the main aspects that should be comprised in response to motivated humanitarian objectives, that is, to assist people who require such assistance, facilitate the return to normality for affected people and communities, and prevent and strengthen preparedness for the occurrence of potential future situations. The main part of this chapter addresses the range of forensic activities that seek to protect lives, to alleviate human suffering, to ensure respect for human beings, and to maintain and protect human dignity and human rights. In that sense, one must take into consideration the main aspects of any humanitarian action, namely humanity, the principles of impartiality, neutrality, and independence. Talking about forensic sciences implies the application of scientific knowledge and methodology for the resolution of legal questions and issues regarding individuals and society. As well known, forensic sciences have a big field of application that include also the humanitarian action in terms of alleviating suffering, saving lives, or protecting health. Reading the documents produced by the UN, ICRC, or listening at the multitude workshop organized by ICRC, the present and future role of forensic sciences in the humanitarian action emerges very clearly. Forensic scientists and practitioners can play a key role in times of armed conflict, in war crimes, in terrorist attacks, in natural disasters, earthquake, flood, tsunami, in case of transport or technological accident, and any other kind of mass disaster. New Jason is actually humanitarian forensics in case. All of these are incidents of mass fatality that usually leave behind a large number of victims that deserve the attention of the forensic community, as do their families. These are situations in which identification of the bodies and their recovery must be a key concern, because the families and population must have the confidence that everything will be done so that the bodies of their relatives are recovered, properly identified, and are delivered to them as soon as possible. Correct? This point is crucial in order for the family to fulfill funeral rituals and to begin the mourning process. In fact, to be informed that a loved one has died is always a personal tragedy, but to remain in the uncertainty of what has happened can cause even greater suffering. In the following section, the main areas where forensic experts apply their specific knowledge, expertise, and methodologies is described and discussed. Heading: The role of forensic sciences and medicine in humanitarian action. Professor Stephen Cordner has often quoted Sergio Vieira de Mello, a Brazilian UN diplomat who worked for UN for more than 34 years and believed that missing persons represent one of the most contentious issues when the restoration and maintenance of peace in a conflict situation is at stake. In 2003, S.V. de Mello indeed said, "My experience has taught me that disappeared are often the most contentious issue in peacemaking. The question that makes contention, bone of contention, on which you fight. The question that makes confidence building all the more difficult. One of the most important and potentially problematic areas of forensic intervention is represented by the identification of dead bodies. In this kind of humanitarian forensic action, it is of utmost importance to differentiate the scenario, that is, where, when, and why the action took place. Dealing with mass disasters caused by natural phenomena is completely different from acting in situations of terrorism or operating in contexts where violation of human rights have been committed. Such differences create heterogeneous scenarios requiring different ways of acting and entailing different possibilities of action. In the occurrence of mass disasters and or acts of terrorism, society usually supports the victims. There is a sense of solidarity and sustenance from the state as well as funds, and the bodies are generally fresh with recent ante-mortem information. The scenario is totally different when working on cases of human rights violations where there is usually no support from the state, or at least no full support from the state, where investigations can take place years after the events, where in many cases one does not deal with fresh bodies but with skeletonized bodies, and where there is a lack of funds. In such a context, there is also a lack of ante-mortem data, and DNA analysis can exhibit technical problems, that is, insufficient biological material for DNA extraction. There is also another specific important field where forensic humanitarian action can take place, that is, those situations of human rights violations with physical and or psychological torture. Everything began with Clyde Snow in 1980s, where he moved to Argentina with a team of forensic anthropologists to identify Argentinian victims of the military regime. Since then, forensic medicine has been used on a regular basis in order to discover evidence of inhumane treatment both on the living and the deceased. Colleagues from the National Portuguese Medico-Legal Institute have recently worked in Mali, carrying out exhumation of mass graves and identifying victims, thereby obtaining clear proof that there were cases of human rights violations, arbitrary execution and torture prior to killing. Furthermore, there was another high-profile case where the Portuguese government decided to send a forensic team to try to recover the bodies of some soldiers who disappeared after the independence of a renowned Portuguese colony. The families had been complaining for more than 20 years that they did not know what had happened to their loved ones, to their military familiars who had remained there, and so a team was sent there to perform the examination of the area and to try to find these bodies, with the result that they were able to recover all of them. All of the areas and sub-disciplines of forensic science and medicine can be profitably applied in the humanitarian forensic action. In particular, forensic anthropology, odontology, genetics, pathology, and the emerging field of clinical, legal, and forensic medicine. In fact, in all of the areas of political tension and conflict, where political and religious violence exists, there is always a tendency for an increase in general violence, such as gender violence, child abuse, elder abuse. Legal and forensic medicine can play a key role in relation to sexual violence, human trafficking, especially in the area of torture and ill treatment in case of detention, which is something that we find a lot of in countries that are undergoing difficult and violent periods. To examine victims of violence or torture, to properly recognize and classify these injuries, and therefore to ensure the safeguarding of lives, thereby promoting changes and permitting courts to fulfill their role, implies the necessity of a specific know-how and expertise in clinical, legal, and forensic medicine. The European Council of Legal Medicine (ECLM) and the European Union of Medical Specialists (UEMS) has approved a denomination and definition which states that legal and forensic medicine involves the observation, documentation, collection, assessment, and scientific interpretation of medical evidence deriving from clinical and or post-mortem investigation. Thus, beside post-mortem investigation, also clinical forensic examinations under different fields of law, that is criminal, civil, work, family and administrative law, have been recognized as a core sub-discipline within forensic and legal medicine. Looking at the content of the residence program in legal and forensic medicine of the main European countries, there is a wide description of what a resident should learn in terms of ascertainment methodology, that is, how to conduct an interview, a clinical examination, how to collect evidence, what kind of complementary examinations to request, etc., and the criteria of evaluation, how to interpret clinical data and other evidences. It is fundamental to know that the doctor-patient relationship changes when you pass from a clinical physician situation to a medical expert situation. Collaboration and trust become something more connected with lack of cooperation and some degree of suspicion, and you have to know how to surmount these problems and resolve the difficulties that pertain to a clinical forensic examination, namely simulation and dissimulation, or more in general, malingering. The clinical interview is crucial. The examiner must be sensitive and empathic, watch out for signs of tiredness or distress, and remain objective. He has to ensure active listening, meticulous communication, courtesy, and genuine empathy and honesty in order to obtain the examinee's trust. He must adopt the right posture and know the questions to use, how to pronounce them, and which questions to avoid, such as leading questions. Individuals may answer with what they think the forensic professional wants to hear. Closed questions, which limit the answer to a choice between yes or no, or too many questions at the same time. The examinee might be confused, or the aim may be to confuse him. Moreover, during the interview, the ascertaining physician has to observe the body language, the facial expression, the tone of the voice, and the gesture of the examinee that can give many clues about the truth of the history and about the consistency of the allegation that are presented. Another essential step of the ascertainment is represented by a systematic clinical examination, which must follow a head-to-toe scheme. Of course, the forensic expert has to be prepared to work in conditions that often are not the best ones. For example, when examining people in the field of humanitarian forensic action, violation of human rights, where sometimes the ambient is not the ideal one but only one possible, then the examiner must be particularly attentive in order to overcome the deficiencies and insufficiencies of the examining conditions. During the clinical objective examination, the forensic physician has to systematically ascertain each portion of the body surface through inspection and palpation, to expose only the part observed in each stage of collection, to examine the clothes very accurately, and to collect physical evidence with a thorough photographic documentation. Sexual assault lo kuda anni ive chepparu. The forensic expert is specifically trained for the examination and description of injuries, describing the number, location, that is anatomical region involved, and the distance from the permanent anatomical marks with a scale, type, that is ecchymosis, hematoma, bruise, laceration, contusion, incised wound, burn, edema, etc. Shape, linear, curvilinear, rounded shape, oval, irregular. Then dimensions, height, width, diameter, depth, use measuring scale. Then orientation, color and surface, that is scaly, crusty, ulcerating, etc. Signs of vital reaction and secondary reactions, that is discoloration, healing, infections, etc. Presence of associated foreign material, soil, sand, wood, etc. ఈ మొత్తం కూడా రేప్లో సెక్షువల్ అసాల్ట్ అన్నీ ఉన్నాయి. Given this basic description, within the field of clinical forensic medicine, the trained physician must also ascertain and describe impairments and damages like reduced mobility, pain, tenderness, and abnormal range of movement. He has to use International Nomina Anatomica and the International Classification of Functional Disability and Health, ICF. రాయాలో అందులో ఉంటుంది classification. In my personal experience, traveling around the world, I have found a lot of colleagues that still perform forensic descriptions using local and national expressions and do not use the international standards. One should know that in clinical forensic medicine, it is important to describe the existence but also the absence of any injuries to have a full and reproducible picture. Documentation in diagrams and photos is crucial, using metrical and colorimetrical scales in order to allow a second opinion or interpretation or reinterpretation to be performed also in a retrospective fashion. In clinical forensic medicine, it is also important to always take into consideration that the situation under observation could have been changed due to the intervention of the emergency team, and the absence of evidence is not evidence of absence. In many cases, forensic examiner intervenes too late when many things are corrupted or disappeared. For example, in the field of torture medicine, many victims totally recover from the physical and psychological point of view, and when they arrive to the forensic examiner, there is no clear evidence of physical and or psychological torture, but absence of evidence is not evidence of absence. As discussed in many workshops organized by the International Academy of Legal Medicine, IALM, and during the recent IALM Inter-Societal Symposium in Venice, 2016, clinical forensic medicine is fundamental in the ascertainment and evaluation of migrants and asylum seekers. The aim is to get evidence as whether the case under examination is really a case of torture or persecution or inhumane treatment or not. The new European Directive 2013 describes a common procedure that should be followed in every European state whenever migrants ask for asylum. A number of substantive changes have been made to the Council Directive 2005. on minimum standards on procedures for granting and withdrawing refugee status. From a medico-legal point of view, it is essential that in every European reality, the same ascertainment methodology and criteria of evaluation are adopted and applied to perform a valid differential diagnosis and arrive at a solid conclusion concerning humanitarian protection. Other areas of forensic action can have a strong humanitarian impact dealing with migrants, asylum seekers, especially forensic genetics. Every day we are seeing migrating families arriving with children that do not belong to those families, which point to the fact that the child trafficking is now becoming more and more common. Children are being kidnapped and used in order to try to obtain asylum, so genetics is also very important in this area for trying to see if the family is really a family. So the role of forensic sciences is today very well recognized. For example, a report on the special role that forensic sciences can play in the investigation and documentation of torture was recognized by the General Assembly of the United Nations in 2007. Given this special role of forensic sciences, all of the major international organizations have forensic groups dedicated to the field of humanitarian forensic action, and even the ICSC, International Criminal Court, decided to create 2 years ago a Forensic International Advisory Board in order to provide advice and expert opinion in all cases with humanitarian implications at a trial. Nowadays, all of the major international organizations use forensic medicine and the forensic sciences on a routine basis whenever they are fulfilling such a role in the field of humanitarian action. It is also interesting to note that international scientific forensic associations are paying special attention to such issues. Perhaps a special prize should be awarded to the American Academy of Forensic Sciences (AAFS), which created the first humanitarian and human rights resource center. This center sets up projects, research activities and training with a basis in humanitarian forensics all over the world. Conclusions. The theoretical and operative roles of the forensic sciences in the present and future humanitarian forensic action can be summarized as follows. Number 1. Forensic experts can provide casework, which means perform forensic ascertainments and evaluations on the field, as already done by ICRC. Second, forensic experts and physicians specialized in legal medicine can promote adequate practices, working and sponsoring this kind of training and capacity building all over the world. Third, forensic scientists and practitioners can help developing guidelines and operational standards, not only of a specific forensic nature. For example, the Minnesota Protocol has just been reviewed in February 2016 and will soon be available on the website of UN. The old manual, written in Vienna in 1989, has been updated with a benchmark for autopsy procedure all over the world, especially where there are humanitarian and human rights issues present. Forensic pathologists have significantly contributed to the revision of this international protocol, which has a strong ethical and humanitarian impact. 4. Specialists in legal medicine and forensic scientists have to stress capacity building. When there are still much to be done, capacity building should include not only forensic practitioners but also all of those who are the recipients and can be directly involved in forensic activities. Capacity building that only takes into consideration forensic practitioners will not be enough, since the participation of prosecutors and judges is essential, as they can learn and come to understand much more effectively what forensic scientists can provide. All of the professionals involved in the humanitarian action must convert the lessons learned and the experiences obtained into effective prevention mechanisms. Through the humanitarian forensic action, the forensic community can promote human rights and attempt to make people more sensitive to all of the rights set out in the Universal Declaration. Everybody knows that human rights are not always a top priority in majority of countries. It is essential to insist, persist, resist and never give up, as depicted by the Acción Poética Escobar, an Argentinian cultural and poetical movement which promotes solidarity. To never give up is of fundamental importance, and I am absolutely sure that in this field of humanitarian action, humanitarian forensic action can really make the difference. Personalized medicine on the basis of biology and computation as a challenge to establish systems biology. The P4 medicine approach was primarily introduced for clinical trial in cancer medicine, involving predictive 4 P's అన్నమాట. Predictive, personalized, preventive and participatory aspects for understanding the biological complexity and better patient care to enhance the voluntary participation of patients. P5 medicine has been supplemented with psychocognitive aspects. అంటే ఇందాక చెప్పిన దానికి సైకోకాగ్నిటివ్ యాడ్ చేశాడు. Psychocognitive aspects to aid patients with personal actions to prevent, cope with and react to illness, decide about therapeutic options and adhere to treatment, enhancing the interaction of patients with healthcare staff. The doctor-patient relationship can be a crucial element to ensure patients' quality of life. Despite the aforementioned principal difference in tasks and obligations between legal and forensic medicine and clinical medicine, the concept of P5 medicine can potentially be introduced into medico-legal and forensic casework from the viewpoint of personalization with clinical relevance to living persons under healthcare management. In medico-legal and forensic investigations on the deceased persons, medico-legal forensic autopsy laboratory systems involving data processing established by the full application of up-to-date medical sciences, including genetics, molecular pathology, toxicology and imaging techniques is essential for the visualization, that is objective demonstration, of post-mortem evidence relevant to clinical observations. In this process, it is particularly important to consider how to learn about life from the deceased, aided by biomedico-legal sciences involving biomolecular approaches. Such innovation in medico-legal or forensic casework will enable the unitary and integrated collection of personalized objective data sets and computation involving ethnic and demographic features to be used to upgrade medico-legal or forensic assessment, leading to juridical, legal and judicial proof with confidence. Interaction with persons concerned may involve conflict in forensic casework in terms of objectivity in the justice system. However, it is important in care of victims or their relatives and criminals in the court process, as well as people's participation in justice system from participatory and psychocognitive aspects of P5 legal or forensic medicine. Furthermore, the computation of massive data, so-called big data, will contribute to the prediction and prevention of hazardous events from diverse continents resides in the rationale, premise and scientific objective of the monograph, both entailed by the neologism proposed by us and that characterizes its title, P5 Medicine and Justice, to which a few words of clarification and explanation must be dedicated. The established and shared concept of P4 holistic and personalized medicine is essentially a return to the past, a rediscovery of the immense humanitarian value of the analogical binomial doctor-patient relationship to which the well-being of the person is entrusted. The P5 extension, in reiterating the value of the past and adding the value of the protection of freedom and dignity of the person, looks to the future through the finalization of the said protection for the attainment of justice, which calls for a personalization combined and consonant with that of personalized medicine. As explained by Zakaria, personalized justice is seeming paradox of a plurimillennial historical parabola that concludes that with the current definitive affirmation of personal and group identity, the entitlement to rights to differences, and the coexistence of anthropological and juridical pluralism in guaranteeing protection equal and without differences of the freedom, dignity and defense of the person, also taking the form of a just trial. A just trial in which, according to Forty and Tarfruser, science and law are divided in their objectives. The criminal law must stick to and lag sciences, not lead it, and the judge must be a consumer, not a producer of scientific knowledge, in order to comply with the principle of legality and human rights, and to build legal foundations sound in science as well as in law. Where again, according to Detmeyer, beyond reaffirming the human rights of Universal Declaration of 1948, and in particular the right to life, liberty, security and presumed innocence, the right to protection from torture, cruel, degrading treatment or punishment, it is necessary to preserve the freedom and independence of forensic medical expert and to establish additional provisions relating to collection of evidence. And as Commande emphasizes, also in order to recognize compensation for personal injury damages and for non-pecuniary harm, characterizing new interests in every legal order where there are adequate resources to dedicate to their protection. In such a context, with the aim of civilization and in line with the tradition and with the view outlined by Pollack, we have always held that to overcome future challenges one has to be aware of the past, and that to form from the past, we grasp the danger of a fragmentation of bio-medico-legal disciplines into separate entities, as well as the consequent indispensability of unitariness, from which naturally arise transdisciplinary innovation and forensic evidence based on heeding other biomedical sciences, and in particular the omic and imaging sciences, upon which parts 1 to 10 of the monograph are focused. An innovative role of the biomedico-legal sciences, perhaps equal to that of genomics, will probably be taken by omics and by living and post-mortem cross-section imaging, which entails significant ethical and epistemological reflections. From the past, we also grasp that forensic evidence cannot but be derived from the combination of research and casuistry and interpretation and criticism, and it is for this that the monograph has been organized into parts, with the final aim of tracing the state of the art of biomedico-legal disciplines and the related innovation and evidence accompanied by juridical and epistemical reflections, specifically concerning forensic pathology and anthropology, clinical legal forensic medicine in the living person, with reference to violence, crimogenesis, humanitarian forensic action related to antisocial behavior, personal injury, sexual assault, torture and asylum, personal damage, malpractice and medical liability under civil and penal law, personal identification and age estimation, forensic genetics and genomics, and toxicology. At the conclusion of this monograph, the scenarios of transdisciplinary innovation of biomedical sciences, biomedico-legal sciences, and in particular their level of evidence have been partly defined in a perspective of further scientific development aimed at meeting the demands of evidence and proof for a better mutual understanding of scientists and judges in relation to personalized justice extensively inclusive of the value of the protection of freedom and dignity of the person. The reader is warmly invited to communicate to the editor any observations, comments and critical issues. Abbreviations: AM Antemortem, DVI Disaster Victim Identification, ICRC International Committee of Red Cross, PM Postmortem, STR Short Tandem Repeat, VNTR Variable Number Tandem Repeat. Introduction. In every disaster or a conflict, the need to recover and identify the deceased is a matter of urgency. Foremost, on behalf of the psychological needs of relatives of those missing, the right to know the fate of one's missing relatives is a fundamental right and should be respected and enacted. This principle holds regardless of the circumstances that have led to the death. Alongside humanitarian needs, disaster victim identification (DVI) also addresses legal concerns regarding everything from civil suits concerning liability in an aviation, train or ferry disaster, the need to provide death certificate for the purpose of inheritance and remarriage, through to the prosecution of perpetrators of war crimes. The scope of DVI operations has evolved significantly since the mid-1990s, when most instances centered on transport accidents involving up to a few hundred victims. Advances have been driven in part technically by the development, particularly in DNA analysis, that have increased the circumstances in which the identification of human remains is possible. Politically, the response to several events of international importance in the late 20th and early 21st centuries, such as conflict in Balkans 1991 to 1999, the World Trade Center attack 2001 in USA, and Pan-Asian tsunamis of 2004, focused a large amount of resource to the identification of the victims. This led to logistical and technical advances in DVI and also to higher expectations from forensic science to be used for human identification following disasters and conflicts, even when there are thousands of victims. First heading: Disaster Classification. A disaster can be defined as an unexpected event causing death or injury to a large number of people. The traditional considerations for activating a DVI response related to mass disasters where external assistance was required following a catastrophic event that overwhelmed the resources of an affected community and rendered it unable to respond adequately acting alone. There are many different types of disasters that can necessitate DVI, including traffic accidents, natural disasters, technical industrial accidents, terrorist attacks, and events that occur during conflict both within and between countries. In addition to the type of disaster, an important aspect when considering DVI is whether it is open or closed. Closed disasters such as aircraft crash are those in which the identities of the victims are known through crew and passenger manifest or a list, although they may not be entirely accurate cases. In such instances, the task of the DVI team is in large match the identity of the remains to individuals known to be missing. In many of these cases, access to relevant ante-mortem information necessary for matching is also facilitated by the relative ease of locating the kin, medical and dental records, and DNA samples. Open disasters are those in which the identity and even the number of victims are unknown. Establishing who was indeed a victim can be quite complicated, even in a thoroughly modern society where it may take several days or weeks, even years to resolve in a situation of limited fatalities. In the case of a natural disaster involving multiple countries or locations within a country and thousands to tens of thousands of victims, multiple nationalities, it can take years to resolve. For victims of conflict, where the number and identity of victims are unknown, individuals disappeared over several years or decades, the location of graves have been obscured, AM records are scant or lacking in their entirety, and political will and monetary resources are slim, identification, where possible, can take decades. It should also be noted that it is possible to have a classification of combined, for example, when a plane crashes on a populated area, killing people on the ground as well as those in the plane. This is a combined class. Then management of disaster victim identification DVI. Interpol recommends that each member state should establish a permanent DVI team with pre-planning and training for disasters. In planning for and following a disaster, multiple facets of the DVI operation have to be considered. For example, initial assessment of the scope of the disaster, mapping and searching of the affected area, estimating the number of victims and the degree of fragmentation of the bodies, methodology to recover and transport the corpses to the lab, methodology to be used for identification, and identifying the individuals and medico-legal institutes that are to be involved in the identification process. The criteria must be met before identification are made, must also be agreed upon prior to bodies being released to families. This becomes increasingly important with increasing number of victims, as this augments the probability of incorrect identifications. Another important decision is the scope of the identification process, that is, with fragmented remains, what will be the operation aimed to identify? Whole bodies only? Recognizable body parts only? Body parts with a specific size only? In case of extreme fragmentation, that is a plane crash, or disruption of the primary burials into secondary and even tertiary mass burials, example in Bosnia, it is also necessary to have a policy in place regarding how relatives of the missing will be informed when a portion of their missing person has been identified. Through this policy, relatives of the missing may, for example, elect to be notified when 1. The entire identification process has been completed and all the remains identified of the individual are collected and returned at a single time in one single piece, one package. 2. The first body part has been identified and they should be informed immediately. Or 3. Every time a body part has been identified, they should be informed and kept in the loop. So this allows the relatives of the missing to assert some control over their relationship to the identification process and also decide at which point they wish to hold memorial and or funeral services. Next, principles of identification. Heading. Identification of individuals killed in mass disasters and conflicts should be held to the same standards as identification of individuals killed or found dead under any other circumstance. The process used in identifying individuals must be robust and the families of the missing must have confidence in the identification process. Decisions regarding the method of identification to be employed are influenced by the condition of the victims, personnel involved in identification, resources available, technology that is used, as well as by the availability of ante-mortem comparative data or items. There are 4 widely recognized means of positive identification, whereby A single method alone can be used to make an identification. All others must be regarded as a means of presumptive identification, which can be used to support identification or generate further leads to identify, but may not in themselves make a identification. Fingerprints. Number 1, fingerprints. 2, dental and medical records including radiographs, that is number 2. Nuclear DNA, that is number 3, and unique medical identifiers, this is the number 4. They are all means of primary positive identification. Visual recognition of tattoos, facial features, birthmarks, scars. Second, a matching biological profile generated by forensic anthropologists by means of age, sex, race, stature, and antemortem trauma and pathology. And third thing is matching medical conditions identified by pathologists, facial superimposition, matching personal effects and clothing, matching identification documents, and exclusion where, for example, there is only one child among the victims, and or matching mitochondrial DNA, mtDNA profiles are secondary means of identification, ideally for use in support of the primary methods. Now let us see the figure 1. Figure 1 is identifications obtained using visual recognition or other customary means are where possible supported by comparing ante-mortem and post-mortem data to allow biological profiling and collection of additional evidence to support the identification. Ideally, identifications should be supported by at least one form of scientific identification, which greatly reduces the possibility of misidentification. SNP based DNA analysis could also provide scientific identification but is not widely used. Scientific identification using odontology would normally require comparison of dental radiographs or other features that would be considered to be unique. Now let us see the process. First item is the leads. The leads are eyewitness reports, event information, then associated documents, identification tags, then visual recognition. These are the leads available. Second stage is biological profiling. In biological profiling or anthropological profiling, you have age, race, sex, stature, healed injuries, and congenital conditions. Now third step is support for identification. If you arrived at some identification above, then how do you support it with documentation? There are unusual features like tattoos, dental work done already, piercings in the ears and nose and other parts of the body, associated clothing, documents and belongings, eyewitness reports and event information. They all go to support the identification made. Then scientific identification finally, if nothing else works, then you have to go to FSL and get scientific identification. The scientific identification consists of fingerprint analysis, then STR based DNA analysis, then unique medical identifiers, and then odontology. These are the 4 things that are used for finally scientific identification. Then methods of identification, now heading objective methods. Objective methods, sometimes referred to as scientific methods, if applied appropriately, can give unambiguous identification. First method, fingerprints. Identification of an individual through fingerprints is a well established scientific method and is routinely used worldwide. In humans, the palm and fingers are covered in skin that is thickened with ridges that helps to grip surfaces, friction ridge skin. The soles of the feet and toes are also covered with friction ridge skin. Friction ridge skin is considered to be unique, and over the last 100 years of application, with millions of fingerprints used for forensic identification, only a handful of issues of misidentification have been reported, making fingerprints a powerful means of identifying. Importantly, fingerprints remain unchanged throughout life, other than through scarring. Several systems have been developed to classify fingerprints, all of which are based on categorization of the overall pattern, that is called loops, arches, whorls, and then identification of distinct combination of features within the above 3 patterns, such as bifurcation, lakes, and ridge endings. Its utility to DVI identification varies from case to case. The remains have to be fleshed and in a state of preservation that allows for prints to be taken. This may be possible even with partially decomposed and even burnt remains. as fingers curl inward when exposed to fire, thus preserving the ridge details. As with all other forms of identification, ante-mortem records also need to exist for comparison. Whether extensive AM records are available varies depends on the country or countries involved. Many countries collect fingerprints from all of its citizens once they reach adulthood, and this information is typically held in a national computer, which allows rapid searching. In other countries, legislation may prevent the storage of fingerprints or restrict it to those arrested in connection to a criminal offence. If no AM records are available, it may be possible to recover AM prints from the personal possessions of the missing persons, assuming there is reliable information on who is actually missing. With a computer-based search using an automated fingerprint identification system (AFIS), a list of candidate matches can be produced in seconds. However, the algorithms are not sufficiently refined to produce exact match, and computer-generated matches have to be examined manually by an expert to establish a confirmed match from the alternatives thrown up by the computer system. Next heading, dental radiographs. The identification of individuals based on the comparison of AM and PM radiographs is arguably a somewhat subjective process, but one which is nonetheless recognized as a scientific means by which a positive identification may be made. While in the past radiographic equipment may not have been available in situ, and radiographers were forced to ship bulky equipment and improvise dark rooms in which to develop the films, this is no longer an obstacle. Portable digital radiographic equipment, radiographic equipment and a portable X-ray, some even handheld, is widely available and rapidly becoming cost effective. If not available in the country in which the disaster or conflict has occurred, this equipment is so portable that it can be carried as a hand luggage in the passenger compartment of aircraft deploying individuals to the scene or experts to the scene. The AM-PM matching of radiographs for identification is more widely accepted in forensic dentistry than in forensic anthropology, where the former is most often based on radiographic image of dental restorations and or implants, which evince the unique characteristics of both the morphology of the lesion and that of the dentist's individual technique, and thus afford multiple surfaces for examination. The latter relies upon matching frontal sinus, cranial suture, vertebral morphology and bone trabecular pattern, not just the orthopedic equivalent of implant fixator, pin, plate, screw and artificial joint. Although the angle at which an AM radiograph is taken may cause distortion, elongation and superimposition of features is in a dental radiograph, the problem is often compounded in an orthopedic image by additional factors such as age-related changes in joint surfaces and trabecular realignment caused by application of differing stresses due to activity level and type with age. In all radiographic comparisons, the years elapsed between when the AM image was made and the individual's death must be taken into consideration. Superimposition of AM-PM images of restorations is a frequently used technique in conjunction with the correspondence of measurements. Again, there is somewhat better correspondence within dental images than even within internal fixators used in the fracture repair, as remodeling of the bone due to weight bearing contributes to the movement of implanted devices over time. Dental records themselves, example notation of teeth treated, conditions of teeth, are not definitive in DVI, as both omissions and mistakes in recording do occur. Such records are potentially useful where comparing large number of AM and PM records to establish possible matches and in providing additional support for an identification. In some contexts, dental records are used where radiographs are not available. The utility of dental radiographs tends to limited to developed countries where individuals typically visit the dentist, and radiography is commonly used as a part of the routine examination or following any major dental work. The dentist normally keeps either the developed radiographs themselves Microfilm of those radiographs are stores digital radiographic images in the patient's electronic file. When disaster victims have not been subjected to trauma that has resulted in disruption of the dentition and AM records are available, forensic odontology is a powerful tool means of achieving identification. Next heading, nuclear DNA. Since its introduction into the forensic science in 1985, DNA profiling has become commonplace in forensic laboratories worldwide. Its primary forensic use is for the identification material recovered from scene of crime. However, it is also widely used in identification of human remains, particularly following on from transport disasters like a plane crash. By the early mid-1990s, the analysis of the STR have replaced VNTR based analysis. STR have several advantages. They could be analyzed using polymerase chain reaction PCR methods, which made them much more sensitive. The alleles of STR loci are also much shorter, making the analysis of degraded material easier. Several STR loci could be analyzed in one reaction and analyzed together using capillary electrophoresis (CE), which greatly reduced the time taken to carry out the analysis. The profiles could be broken down into series of numbers corresponding to the number of core repeats present in each allele, which allows comparison to be made between DNA profiles generated within a laboratory and also to compare profiles generated in different laboratories. STRs are found throughout the genome and they are composed of the short blocks of sequence that are repeated in tandem. The STRs commonly used for forensic analysis have blocks of sequences that are 4 or 5 BP long. While there are numerous STR loci in human genome that could be used as forensic markers, around 20 to 25 only are commonly used by forensic scientists and FSLs. These have been selected for being highly discriminating, that is typically having a large number of variants, alleles, that differ in the number of core repeats and for being easy to analyze using PCR based technology. Using current technology, DNA profiles can in principle be generated within a few hours and produce a profile that is extremely rare if not unique. Because of the limited discrimination capacity of early profiling systems, it was not until 1991 that DNA profiling was successfully employed in the middle of human remains. In this case, a murder victim in the UK had since been successfully applied to cases that range from identification of individual body to more complex scenarios with multiple victims following air crash, fire explosion, terrorist attack, natural disaster and conflict. A distinct advantage of DNA analysis in DVI in UK is that all tissues contain DNA and therefore the method will work on highly fragmented remains and enable fragments of bodies to be reassociated. As with the other form of identification, the use of DNA analysis in DVI will vary from context to context. Whether DNA can be recovered from the human remains depends on the degree of environmental insult. For example, if the remains have been subjected to a high temperature in excess of 200 degrees centigrade or there has been a long delay in locating and recovering the remains, the DNA can be highly degraded. It may not be possible to generate a profile. AM data is required for comparison. This can be in the form of direct comparison. Direct comparison if a biological sample from the missing person is available, or more commonly by comparison to biological relatives of the missing person. Ward sample gaani or relatives gaani. Okay. Next heading, unique medical identifiers. On the occasions, PM are repeated in tandem. The STRs commonly used for forensic analysis have blocks of sequence of 4 or 5 BP long. Well, these are numerous STR loci in human genome that could be used as forensic markers. Around 20 to 25 are commonly used. Next. These have been selected for being highly discriminating, typically having a large number of variants alleles that differ in the number of core repeats, and for being easy to analyze using PCR based technology. Using current methodology, DNA profiles can in principle be generated within a few hours and produce a profile that is extremely rare if not unique. Because of the limited discrimination capacity of the early profiling systems, it was not until 1991 that DNA profiling was successfully employed in the identification of human remains. In this case, a murder victim in UK. It has since been successfully applied to cases that range from identification of individual bodies to more complex scenarios with multiple victims following, for example, air crash, fire, explosion, terrorist attack, natural disaster, and conflict. A distinct advantage of DNA analysis in DVI is that all tissues contain DNA, and therefore the method will work on highly fragmented remains and enable fragments of bodies to be reassociated. As with other forms of identification, the use of DNA analysis in DVI will vary from context to context. Whether DNA can be recovered from the human remains depends on the degree of environmental insult. For example, if the remains have been subjected to high temperatures in excess of 200 degrees centigrade, or there has been a long delay in locating and recovering the remains, the DNA can be highly degraded. It may not be possible to generate a profile. AM data is required for comparison. This can be in the form of a direct comparison if a biological sample of the missing person is available, or more commonly by comparison to biological relative of the missing person. Next heading, unique medical identifiers. On occasions, PM analysis of human remains may happen upon a medical implant. Typical examples are artificial joints and pacemakers. These medical devices come with a unique serial number and can allow for unambiguous identification. As long as the relevant information can be located. Needless to say, identification through this method are not common. Data collection. Heading. Data collection, both PM and AM, is a critical component of DVI operation. Several agencies, both national and international, have developed forms that facilitate the systematic collection of data that can be used in identification process. The most commonly employed system for DVI work uses the Interpol forms. They have been specifically developed for DVI work. The International Committee of Red Cross (ICRC) has also developed AM and PM forms which have been designed for use in post-conflict scenarios. Both these systems can be computerized with the data entered into a relational database, allowing AM and PM data to be cross-searched. National authorities have also developed their own systems. For example, in US, Disaster Mortuary Operational Response Team (DMORT) has its own set of recording forms and database recording similar information. Obviously, the more victims and the greater the fragmentation of those individuals, the more imperative it is to computerize PM and AM records. Searching by hand through records for matches, especially when the personnel who originally recorded PM details are no longer available and memory is lacking, is a frustrating exercise. Post-mortem. The cooperation and teamwork of experts, sharing of information and agreement concerning the adherence to criteria by which an identification will be made are critical to the management and effective performance of any DVI team. The workflow in the morgue and hence floor plan should be arranged to facilitate the processing of remains as well as the communication among the various team of experts. The technical experts involved in the collection of data will vary depending on the state of human remains. Typically, teams will comprise some or all of a pathologist who will undertake a PM examination, an anthropologist to examine the skeletal materials, an odontologist to take radiographs and examine teeth, a radiographer to take whole body X-rays, a fingerprint expert and a biologist to take samples for DNA analysis. Other individuals, for example, police officers, crime scene investigations, will also be involved carrying out tasks such as documenting personal belongings, clothing, taking photographs, ensuring chain of custody for any evidential items. Ante-mortem. A prerequisite to the application of any of these methods is the existence of relevant, reliable AM data. Following a mass disaster, AM data collection interview is typically conducted with relatives of the missing. The collection of AM data is as much a specialized process as conducting a PM examination, and interviews of the relatives of missing should be performed only by trained personnel. Interviews are best performed by native speakers of the language, so in the country of training of personnel should be planned. During the interview, information concerning the individual's physical description, a description of the clothing the individual was wearing, and any personal effects that were with the individual are recorded on an AM data form. This form should be compatible with the PM form to allow searching of comparable fields of information. A photograph of the missing person is collected and the existence and whereabouts of the medical and dental records for that individual are noted so that they can be requested from doctors and dentists at a later date. Photographs of the victim smiling can also be useful as the teeth may have distinguishing morphology or features such as spots caused by hypoplasia. During this interview, if possible, DNA samples are taken and latent fingerprints are enhanced and collected from the home of missing person. DNA samples may be direct reference samples, example from the missing person's toothbrush or hairbrush, or indirect reference samples, preferably from the immediate kin, father, mother and children. The above methods work very well in developed countries, and the process above describes the situation in a country with well-developed infrastructure. However, the infrastructure of many less developed countries does not facilitate the collection, maintenance and storage of such relevant records that drive the DVA process for the citizenry of developed countries. In post-conflict situations, even where such records may have existed at one time, they may have been destroyed during the conflict or its aftermath, or have become unavailable due to political and boundary shifts in natural and other mass disasters in less developed countries. The same may be true regarding destruction of extant records or the lack of relevant records. Furthermore, the subsequent to mass disasters and conflicts, many individuals are frequently displaced internally or have become refugees in neighboring countries. This makes locating relevant kin of missing difficult and organizing and conducting AIM interviews challenging. Whenever possible, it is recommended to avoid re-traumatizing the relatives of the missing by repeated visits and questions, as these doubtless reopen wounds, doubts and raise anxiety. Next heading, selection of methods for identification. In any mass disaster, the method that can be employed successfully will be dictated according to the condition of the remains, the composition of experts in the DVI team, technical and financial resources available, and the choices made by the team's leadership during course of its operational mandate. Prior to DNA analysis, comparison of dental records was the most widely used method to achieve identification. Analysis of 5 disasters in the UK between 85 and 89, which comprised 3 air disasters, a capsized ferry, a fire on an oil platform, identified that dental records have been used to identify on average over 80% of recovered bodies. In more recent large-scale disaster, for example, World Trade Center investigation, DNA was the first choice for identification and generated the majority of identification by a single modality, 86%. Dental and fingerprints combined only yielding additional 10%. Similarly, since the inception of the International Commission for Missing Persons (ICMP) DNA lab in Tuzla, Bosnia, identifications from 92 to 95 Bosnian conflict have been DNA-led, with secondary means of identification used where possible. To support the DNA-based identification, as of mid-2011, over 13,000 individuals in Bosnia-Herzegovina have been identified and returned to their families. In contrast, although DNA was initially a method of first choice in the Asian tsunami identification in Thailand, 11 months after the tsunami, 80% of foreign victims have been identified using dental comparisons. Dental analysis with local victims identified 55% of Thai victims, lower than with foreign victims due to lack of AIM records in many cases. Overall, in the 15-month following disaster, identifications have been made using dental alone, 1105, dental plus others 1451, fingerprints alone 670, fingerprint plus others 927, DNA alone 27, DNA plus others 459. There are reasons for the success ratio of identification methods in each of the above examples. In World Trade Center, victim fragmentation was extreme and the goal was set to identify every piece of tissue greater than or equal to 5 cm cube, thus necessitating the use of DNA. In Bosnia, lack of AIM medical and dental records commingling in secondary mass graves and time elapsed since the death of the victim are but a few of the factors that dictated the reliance on DNA rather than other primary identifiers. In Thailand, important factors included dental-based identification could be made for many of the foreign victims relatively rapidly. Many bodies were not fragmented, removing the requirements for DNA-based reassociation. Fingerprint analysis was successful with many Thai victims. Limited in-country facilitations were capable of handling the volume of DNA samples from the disaster, and decomposition of tissue was rapid in tropical environment, making DNA analysis difficult. Evaluation of identification data. The exact procedures used in the identification process vary from country to country and from scenario to scenario. Typically, identification are made through an organization that acts as an identification commission on board. This would typically include an expert from the main disciplines involved in making identification, like investigator, crime scene investigator, fingerprint examiner, odontologist, pathologist, anthropologist, DNA analyst. Should be presided over by a legal authority charged with issuing death certificate. In this manner, all the evidence contributing to the identification of an individual in a mass disaster is reviewed a final time by everyone who has contributed to that process, and a legal decision concerning the establishment of positive identity and repatriation of the remains of a kin can be completed in time. Evaluating the identification is a complex task, especially as the number of victims increases. This heightens the possibility of coincidental matches, which, if not detected, can lead to misidentification. As often cited example where this occurred is the identification of a fire officer recovered from World Trade Center following 9/11, who was misidentified on the basis of his clothing that identified him as a fire officer from a particular unit, coupled with a rare congenital anomaly in spinal vertebra. The identification was not confirmed by any of the recognized objective methods of identification, and DNA analysis later exposed the misidentification, but only after the remains had been returned to the wrong family. The rare medical condition has been shared by one of his colleagues in his unit. Such seemingly remote coincidences become more likely as the number of victims increases. Conclusion. The requirement for government and indeed international community to be able to mount an effective DVI response following disasters and conflicts is widely accepted. Recent technological advances in terms of DNA analysis, portable radiographic equipment, sophisticated computer databases that allow comparison of AM/PM data have increased the number of cases where the identification of human remains is possible. There will always be limitations as to what is possible. For example, following WTC identification, even with an enormous effort expended, no identification could be made for over 40% of the victims, which should not distract from the success in identifying over 1500 victims. With continued improvements in technology, preparedness and infrastructure to deal with a large number of samples in a timely fashion, there is the potential to have a greater proportion of victims identified and returned to their families following future disasters and conflicts.